Hello and welcome back everybody. In this video we are going to talk about eukaryotes and prokaryotes, mainly the differences between them. So if we look at the term eukaryote, so we can find clearly distinctly two parts of the word, that is the u part and the karyotes part. Okay, so u means advanced in biology, and karyotes refers to the karyon or the nucleus. Okay, and the pro refers to early or before, and the karyotes again refers to the nucleus. So if we join these two terms, we get the eukaryote meaning as the advanced or developed nucleus, and the prokaryotes refer to the primitive or before the development of the nucleus. So that is the first difference, as we can see, which comes directly from the word, that is the nucleus is present in case of eukaryotes and absent in case of the prokaryotes. Now what is the second difference? We can say that the number of chromosomes present in eukaryotes is usually more than one, but in prokaryotes it is one, and there may be presence of plasmids in the prokaryotes. So what is plasmids? So plasmids, let me draw a prokaryotic cell over here. So then let's say that is a cell, and let's say that this is the genetic material of the prokaryote. A plasmid is an extra piece of genetic material. Let's say this is over here, extra piece of genetic material which the cell can take in it into itself by the process called transformation. So if the cell is in a medium where this genetic material is free in the medium, then the cell can take that genetic material. If it belongs to the same species and cell finds it useful, then the cell can take it inside to the process called endocytosis, and after taking it, it will be a part of the genetic material and it will be called the plasmid. That is the extra genetic material. So that one, that extra piece of genetic material, that is the main genetic material, this is the extra genetic material, and this extra genetic material is called the plasmid. Okay, so that is the plasmid. Okay, so this plasmid is present in case of prokaryotes and absent in case of eukaryotes. Okay, so we don't take genetic material like that inside our cells. Okay, so that was plasmid, and that plasmid can provide resistance to antibiotics to this prokaryotes or the bacteria. So for the bacteria it is very important to be resistant to antibiotics, right? So that can be provided by plasmids. So it is not present in all types of bacteria, all types of prokaryotes, but some types of prokaryotes have this plasmid and it does the function as mentioned. Okay, so that was the second point of difference. Let us see the third point of difference, the genetic recombination. So what is genetic recombination? Genetic recombination refers that how the genetic material is getting changed from generation to generation. Okay, so if the genetic material is not getting changed, then the evolution will not occur, and if evolution doesn't occur, then that species is endangered. Endangered means the nature is changing, and if we don't change ourselves according to the nature, then our survival is very difficult. So that is the principle of evolution, and this eukaryotes, this genetic recombination is done by meiosis, as we will study in the cell division chapter, and by fusion of gametes. So as we know, we have two chromosomes, one from our mother and one from our father, and these two sets of chromosomes have some genes which may be dominant or recessive, and with the combination of those genes, different combination of those genes can help in genetic recombination also. So prokaryotes or bacteria also has genetic recombination abilities. It can also change its genetic recombination. One of them is the transformation, as I told, taking in of the plasmid. The transduction is the genetic recombination procedure, which we will discuss in some other video. It is basically the changing in the genetic material when some virus attacks the prokaryote or the bacteria, and then with the help of the virus the bacteria can change its genetic material. When I will make the video, I will put the link over here somewhere on the screen. You can click on it and then learn more about transduction. So conjugation is another such process. It can be called the sex in bacteria. So bacteria can do sexual reproduction through this conjugation, by which in conjugation it forms pili, and one bacteria called the F plus bacteria, which has the F plus plasmid, can transfer its plasmid to the another bacteria which is the F minus, which doesn't has it, through the conjugation tube. So that will be also a discussion for separate video. So that was the difference between eukaryotes and prokaryotes on the basis of genetic recombination. The next point is that the membrane bound organelles, for example the centrosome, lysosome and Golgi bodies, the endoplasmic reticulum, the mitochondria, the nucleus, as we talked about, the plastids, all are absent in the prokaryotes and present in the eukaryotes. So that is a great bit of difference. Many places you will find that this Golgi body absent present means present absent in prokaryotes, then centrosome eukaryotes present, prokaryotes absent, but it can be all generalized under membrane bound organelles, which will be easier to remember, which are absent in case of prokaryotes and present in case of eukaryotes. But it has some exceptions such as vesicles and vacuoles, which are present in case of both. So if you consider vesicles and vacuoles to be organelles, and they are obviously membrane bound, so they are present in case of prokaryotes and eukaryotes also. So that one. And the next point goes that the DNA wrapping on proteins, so it is with the help of histones in eukaryotes and other scaffolding proteins in case of prokaryotes. So what is this? Let us again learn about this in short. Where is my cursor? Yeah. So if we usually look at the genetic material, if we stretch the genetic material, we will find that the genetic material is very long. Genetic material can be as long as up to 6 meters. And how can we fit such a large genetic material into such a small space called the nucleus, which is 0.1 millimeter or smaller than that? Okay, so for that we need to fold or wrap our DNA around some special proteins, and that protein in case of eukaryote is the histone protein. So let us say there is some histone protein. Let me increase the size. So let us say these histone proteins, I am not going into structure. These histone proteins are present in the eukaryotes. So this histone proteins in the nucleus has the genetic material of the bacteria, not the eukaryotes, wrapped around them. Okay, so here the genetic material enters and it is wrapped, and then sorry, let me redo it. Like this enters, wrapped around, there we go, like this, like this, and there. So this long genetic material, let's say that's the genetic material, unwrapped or unfolded genetic material, that was. So if we fold it around this histone proteins, it becomes a little bit thicker and shorter, right? So that's not the final way of folding. Then this structure which is formed is again folded and again folded and again folded, and by those foldings we can fit this structure into a small nucleus, so that this this protein with which the genetic material is folded around or wrapped around is the histone. So this protein is the histone, which is absent in the prokaryotes. So if it is absent in the prokaryotes, how does the prokaryote fold its genetic material? Actually, it does it with other scaffolding proteins called the non-histone proteins. But the histone protein is absent in the prokaryotes. So that was that point. Let me again remove this, that one also. Now the ribosomes. The ribosome in eukaryotes is 80S type of. S is the Sevtberg unit if you have studied, and the prokaryotes it is 70S type of. If you look at the subunits, it is 60 plus 40. Larger subunit of the ribosome is 60S, and the smaller one is 40S type. And in the prokaryote, the larger one is 50S type, and the smaller one is 30S type. Okay, so that will be also uh, discussed about in detail in some other video, which may have the link may be given in the video. Okay, so that was about the ribosomes. Flagella and cilia, the arrangement is 9 plus 2 arrangement, and in prokaryotes it is 9 plus 0 arrangement. So what do we talk about? Let me discuss this also. Okay, so the flagella is a locomotive structure present in bacteria and prokaryotes and eukaryotes both. Okay, so that structure which moves and helps the cell to move around in water, and cilia are structures which are smaller than flagella and present like hair-like structures like this. So these structures are the cilia, and they are the smaller than flagella, but they also help in locomotion, okay, in the water. So this flagella and cilia can be present in both eukaryotes and prokaryotes, but their structure is different. So if we cut a cross section and zoom it over here, we will find somewhat like this. This is the zoomed version of the cross section of this flagella. There will be a central microtubule, that is which comprises of two microtubules actually, two microtubules over here, and then it will be linked with 9 rows of these two microtubules like this. It will be linked. Okay, so it is very rough diagram. This will again require another video for clear explanation. Okay, very roughly I am drawing like this. There will be 9, and this individual circles are rows is a microtubule. Okay, so in the eukaryotes this central structure is present, but in the prokaryotes is not present. So if there is nine such what do we say, microtubule groups or pairs, so it is 9 plus the central 2 is 9 plus 2, but in the prokaryotes it is 9 plus 0. So that is the difference between the eukaryotes and prokaryotes on the basis of the cilia and flagella structure. Okay, so the next over here is the cell wall, and as we all know that the plant cells and the fungi cells possess the cell wall. Plant cell has the cellulosic cell wall, and the fungi cell wall is made up of chitin as many may pronounce, and prokaryotes also contain cell wall which is called capsule or the cell wall, and maybe the slime layer, which many may not consider as the cell wall, but is outside the cell membrane. So that two structures may be present, and it is of complex proteins, complex carbohydrates, sorry. So very complex structure of the cell wall of the prokaryotes, and the cell size in eukaryotes is 10 to 100 micrometer, and in prokaryotes is 1 to 10 micrometer. So you can imagine the eukaryotes to be 10 times the size as of prokaryotes. Example can be plants and animals, and also can be fungi and protista. But in prokaryotes, the example is bacteria and archaea. Archaea are the organisms which live in extremely hostile environments, like the extremely high temperature environments and extremely saline environments, etc. So that was the difference between eukaryotes and prokaryotes in a nutshell. Thank you.